Yeah, thank you, Anna. Hello, everyone. It's my pleasure to be here. So as Anna said, I will be talking about biostatistics, some concepts, and with a focus on hypothesis testing, and I brought an example data set from transcriptomics. So let's start with the data and why we need biostatistics. I think you're all aware that we live in times where we have increasing amounts of data available. So we have different omic techniques, we have imaging data, we have clinical data, and often different types of data are even combined in the same experiment. So in this transcriptomics example that we'll see today, we have about 20,000 genes from 32 samples, which can be represented in a kind of matrix. And even though this is a comparably small data set, it still turns out that human brains are not particularly good at dealing with matrices of that size, right? So that means we need ways to make the data accessible to humans and also ways to trust the results. In addition to there being lots of data, the data also have often come a long way. So here you see the experimental pipeline for transcriptomics, which I'll talk more about later. But for now, I just want to point out that data analysis, and in particular statistics, is often seen as the end of that pipeline. Because if you read a statement that says there's a difference between the conditions, it's not trusted unless it reads there's a significant difference between the conditions and it has some p-value attached to it. So I'm a biostatistics consultant at EMBL, and I discuss a lot both about small and large data sets with biologists, and I thought I would just share some doubts and questions that I deal with on a daily basis in my work, which is, why is it the p-value that we trust in, and should it be? Why do we have these clumsy definitions in statistics? Why does the result seem to depend on which particular statisticians we ask? And if we're talking about big data sets, can't we just go and interpret anything into them? So if these are your questions, stay tuned. I'll come back to them later in that talk. First, I wanted to tell you that statistics is more about than just p-values, right? It really is a large toolbox with tools for, yes, scientific hypothesis testing, but also visualizing data and estimating biologically relevant quantities, as well as finding patterns in the data. So what a lot of statistical methods have in common is that they try to reduce the complexity of the data. For example, we use principal component analysis for reducing the dimensionality of the data. We often report averages, like the average methylation level of a genetic region, rather than the individual sample methylation levels. We also report p-values, which are a summary of the data. And if we test lots and lots of genes, what we often want is a reduced candidate list of genes that we can then further work with. So while reducing complexity is great for our brains and helps us understanding the data, it also means that we're losing information. And what I'm showing here is the Anscombe quartet. So that's a data set that consists of 4 smaller data sets. Both have X and Y. And I think what you can see quite clearly is that they show qualitatively different scenarios. So on the left, you have a scenario where Y seems to be linearly dependent on X, and on the other side, you have somewhere where Y doesn't seem to depend on X, but there is a huge outlier somewhere. But the thing is, if we look at the mean and the standard deviation and the correlation coefficients, they are all the same in all the 4 data sets, which means that we really shouldn't blindly trust this kind of summary statistics, nor should we blindly apply them. So this is already my first lesson for today. If you can, please try to get a look at the data themselves. Right, so what I've shown you here was summary statistics, which means they describe a certain sample that we have. But what most of the time we're after is so-called statistical inference, which is we want to draw conclusions on some bigger population based on that sample. So here's a very silly example. Imagine that you want to make a statement about all apples of one brand and how many worms they usually have. What you will probably do is you will pick a few bags of apples from that brand, maybe 10, and then by this you generated a sample. From this sample, you count the number of worms that you find in there, and you will want to make a statement about this bigger population. So you want to perform inference. So inference always involves assumptions, and these assumptions, not always, but very often, come in the form of statistical models. And the idea here is that we have a data generating process in reality, which involves the way in which you choose the bags of apples, and maybe you have some splicing machine where you can pour the apples and it will splice them and spit some count out. So that's the data generating process. And then on the other hand, we have the model, which I try to depict with these kind of glasses. So this is our view of the process, or our view of the world. So a statistical model tries to represent the data generating process with equations. It summarizes a set of assumptions. So for example, we could say that every bag has the same size. It also specifies relations between variables. For example, we could say that the age of the apples increases the number of worms that we would expect in them. And the important part is that the model can help us to calculate the probability of the data under the assumptions that we make. So why is that useful? I brought another example for this. So let's imagine we have a disease which has a known prevalence of 4% in the population. And now we have a test group of 100 persons that have a specific precondition. And each of these persons gets tested for the disease. And within these 100 persons, we find 9 that have the disease. So is that surprising or not? Well, we don't know unless we can put some probabilities on the outcomes. And this is exactly what we use a model for. So here a binomial model would apply. It's rather simple. It just says that every individual person has a probability of 4% of having the disease. And by this, we get the probabilities of 0, 1, 2, and so on, persons with the disease in the test group, under the assumption that the prevalence is really 4%. So we would expect 4, right, out of 100, which equivalents to 4%, but we observe 9, and now we have a probability associated with it, which is 2%, and it's rather small. So we might conclude that these kind of data are unlikely under this assumption of 4%, and maybe the prevalence is really different in our test group of persons that have the precondition. So there is a famous quote by George Box. It says, "All models are wrong, but some are useful." So is our model wrong or useful? Well, I would argue both, because it's most certainly wrong. The model is oversimplifying. Models are always oversimplifying. In this case, factors like age, region, and other preconditions are neglected by the model. Also, these assumed 4% come out of nowhere, while in reality, they have hopefully been measured somewhere, and that means that they are also subject to measurement uncertainty. So we could say the model is wrong and we don't use it, or we can find that the model is still useful. And I think it is, because it gives us a straightforward estimate of how unusual the data are. And this can be a starting point for further investigation. So in this case, we can use the binomial test, which is based on the binomial model, which will tell us that when the true prevalence is 4%, the probability of observing an event or a count of 9 out of 100 or more extreme is 2%, which is very low. This 2% is a p-value, and it might not be exactly accurate, but it's still a rough idea, and you can decide that this is low enough so that you will want to further investigate the precondition that this group of persons has. Okay, so with these ideas in mind, I think we're ready for the RNA-Seq example that I brought. So we are going to look at a study where the authors were looking for pathways that are associated with the outcome of an RSV challenge. So RSV is a respiratory syncytial virus, which is a common cold virus. In this study, there was a test group, or some adults who volunteered, and they got a nasal swab before doing anything else, which went to RNA sequencing. So there's nasal tissue as samples. After this, they were all challenged with the virus. So they all got some drops of the virus in their nose. And after a couple of days, some individuals get the cold and others didn't. So the question was now whether there are genes that were differentially expressed in the cold group before the exposure, or in other words, whether there are some genes that are predictive for the outcome of the challenge. So In this study, there were 32 samples, and for each of the samples with the nasal cells, the RNA was extracted, prepared for sequencing, sequenced, and then there were sequencing reads that were mapped to the genome. I think this is the important part for today. So that means for a genome region of interest, for example, a gene, where we have a number of reads that map to that region in each sample. And this gives us the following table that we're discussing now. So this table has 20,000 rows for 20,000 genes and 32 columns for 32 samples. And in each of them, there is a count, which tells us how many reads from that particular sample map to that particular gene. In addition to that, we have metadata on the sample, and this metadata tells us more about the samples. And the important part here would be, for example, whether this sample belonged to a person that developed a cold or not. So this means by this information, we can divide the samples into a cold and a no cold group, and then we can compare the number of reads in each group to each other. So here I brought an example gene where I'm showing the normalized counts for. So they are normalized to the respective sample size. Each black dot here belongs to one sample, and I split them by no cold and cold group. I'm also showing you the averages of the normalized counts, and you can clearly see that they are different in the two groups. But at the same time, there is also variation in each group. So the question that we will want to answer is how likely it is to see this kind of difference just by chance if in reality the expression of that gene had nothing to do with the response to the viral exposure. So if we're asking how likely it is to see something by chance, what we're after are probabilities. And the way we can get probabilities is by using a statistical model. So here's one of the most common models for gene expression. It says that the expression of a gene is described by an average baseline expression of the reference group. And here the reference group would be the no-cold group. Then there is a difference between means, which could be 0. We are going to test that. There's some typical variance that we would expect for count data, and some additional variance called overdispersion that is due to biological or also technical heterogeneity that we're dealing a lot with in this kind of biological experiments. So it said the difference between means could be 0 or not, and we want to test it. So that means I'm actually talking about two models here. There's one model called the null model that says there is no difference between the groups, and the data could as well be described by one overall mean. And the other is called the alternative model, which says that the groups are different or something is going on in the data for that gene. So these models belong to two hypotheses, the null and the alternative hypothesis. And the question is, how do we decide which model or which hypothesis we believe in? And the way we go in statistics is that we assume the null model unless we have enough evidence against it. So for you to remember this, you can now imagine me in 20 years. I will be old and I will be grumpy and I will be wearing my null hypothesis glasses. So that means I don't believe in new scientific findings unless you show me some data that hold enough evidence against my null hypothesis view of the world, right? So evidence means the probability of your data under the null model is very low. And in practice, what we do is this. We calculate the probability of the data under the null hypothesis by summarizing the data into a test statistic. So in this example, the test statistic is called Z, and it gets large when the data deviate from the null model. So what we do here is we compare three quantities of the data. And this is also the case in other kind of tests. So we're comparing the variance. So if the variance within each group is low, then this increases the test statistic. If the sample size is large, then it increases Z. And if the effect size, which is the difference in means, is large, this also increases the test statistic. Yeah, so it's three things that go into there. And what we also have is a null distribution, which gives the expectation of our summary of the data under the null model. So we can either get this null distribution from mathematicians, or we could also get it by simulating data and calculating Z under the null model again and again. Finally, we get a p-value by comparing Z to the null distribution. So the p-value is the probability under the null to see a value of Z at least as extreme as the observed Z. And to show you what we do graphically here is that we compare the tails, the extremes of the distribution that are larger than Z to the overall area of the distribution. And in the end, we make a decision. So we reject the null hypothesis if the p-value, the probability of the data under this null hypothesis is below some cutoff. And this cutoff is called significance level. Okay, so if we apply this kind of procedure to the data that I've just shown you, we will get a very low p-value. It's 0.0001. So that means with the common significance level, so a common one would be 5%, but that's just a convention. We could conclude that this gene MXD1 is differentially expressed between the code and the no-code group. Okay, so I hope what you have learned from this example is that the p-value quantifies evidence against the null hypothesis. So this is the intuition that I want to give you on your way for the p-value. Yes, it has a very clumsy definition, but it also has, I think, a very clear intuition. And with that in mind, I think we can now skip ahead a bit in the publication. And of course, not only this gene that I have shown you was tested, but also 20,000 other genes, and based on some thresholds, some of them were called differentially expressed and others were not. And for those that were differentially expressed in the col group, so higher expressed in the col group, the authors found that unusually many of them had something to do with the biological process that in turn is associated with neutrophil activation. So that was a new hypothesis that maybe neutrophil activation is associated with the outcome of the challenge, and that led to follow-up experiments. So here they looked at nasal protein levels now, protein, not RNA, of one neutrophil-associated mediator called MPO. And they claimed that these protein levels were significantly higher in the cold group relative to the no-cold group. And we get a p-value for this. So what I'd like to do now is assume you read this kind of statement in a publication. Let's dissect it. What do we learn from this? Well, how do we interpret the p-value? This is what the p-value tells us. It tells us that under the assumption that the test makes, and under the assumption that there is no difference between the protein levels in the two groups, the probability of seeing the data that the authors saw is low. It's only 1%. So if you were wondering why results sometimes seem to depend on which particular statisticians you asked, well, one answer might be that maybe you phrased your research question slightly different in the two cases, but also another important answer is that, well, the assumptions matter, right? And it's always good to question the assumptions and ask yourself whether they are somehow reasonable. So in this case, the authors used the Wilcoxon test. This is a very popular test because it makes very little assumptions on the data. So there's also little to argue about. The Wilcoxon test, for example, doesn't have a statistical model in the background. The downside of that test is that, well, if you assume little on your data, it's also hard to get surprised by your data, meaning that the Wilcoxon test has a lower power than other methods. So it is worse, or it's not as easy to detect something with the Wilcoxon test as with other tests that use a statistical model in the background. So still, we might worry about this clumsy definition about the p-value, and I admit it's very clumsy. And I sometimes have the impression that people would be happier if the p-value could just be interpreted as the probability of the null hypothesis being true. So the problem about that is that if P was the probability of the null hypothesis, then we could in fact prove anything by just collecting very little data. Because remember that we start by assuming that the null hypothesis is true. So you could set up a completely arbitrary null hypothesis, just show 2 or 3 data points. These 2 or 3 data points will not have enough evidence against the null hypothesis. And then you could say, I knew it, I proved the null hypothesis. So, I mean, this is an exaggerated example, and I'm sure you noticed that this way of argument doesn't make too much sense, but I think the message is also clear. Little evidence against the null hypothesis doesn't mean that the null hypothesis is true. So that means also that this clumsy definition is for now the best that we have. Why is it the p-value that we trust in, and should it be? Well, I hope I convinced you by now that the p-value is a useful summary of the data because it quantifies evidence against the null. But of course, it's still a summary of the data, and we learned earlier on that when summarizing the data, we also lose information. So here I made up 3 different scenarios where we're comparing the no-code and the code group. And all these 3 comparisons have a very similar and very low p-value. Now, but due to different reasons. So in the scenario on the left, we really have a substantial difference in expression between the 2 groups, which makes the p-value small. But in the middle, we have not that large of a difference in average expression levels, but we have lots of data points that give evidence for that difference being real. And in the end, we have like a very tiny difference in means, but also a very small variance, which also reduces the p-value. So the lesson here is that a p-value alone will not tell you a lot. I think if you have a p-value, you should always also look at the effect size at the same time. Because remember how the null and the alternative hypothesis are defined, right? The null hypothesis says that the difference in expression is 0, exactly 0. And the alternative hypothesis is really anything else. So that means if we had the tiniest fold change or expression change, but lots of data to resolve that one, we would reject the null hypothesis. And now have a look at this. This is the transcriptional regulatory network for E. coli. And I think it's clear that in this network, really everything is connected to everything else, right? So now imagine that you have some condition that changes the expression of a set of genes in that network. Would you really expect that this change in expression in the set of genes causes no change at all in the remaining genes? Well, probably it does, but the changes won't be that large. So you will not be interested in finding that all the genes are somehow differentially expressed, but you're indeed interested in those genes that have a substantial expression change. So here at this point, I repeat my point. Please also have a look at the effect size. So for the example that I brought here, we don't get an effect size reported, but what we do is we get a chance to look at the data themselves. So here you see the protein levels in the cold and the no cold group, and I would guess by eye that maybe the levels in the cold group are about 5 times higher than in the no cold group. And to be honest, I have no idea what that means for protein levels, if this is high or low. But at least it's 5-fold and not 1%. So we had this last questions, whether we cannot just interpret in anything into the data sets if they are just big enough. And I do admit that there is a danger to that. But I also think that we have good practices that should keep us from doing so. For example, we should publish the data and we should make the results reproducible. And there are additional ways of quality control, one of which I would like to highlight today, namely adjustment for multiple testing. So in this study, the authors found 80 genes that were differentially expressed, but this differential expression was not based on a p-value cutoff, but on an adjusted p-value cutoff. So what's the difference between p-value and adjusted p-values? For this, let's come back to the scenario from the beginning where we compared the counts of persons with the disease in a test group to a hypothesized prevalence. And here we found, using a binomial test, that the prevalence in the test group is significantly different from 4% with a low p-value. And this test built on a binomial model. Just as a reminder, this model assumes that every test person individually has a probability of 4% of getting the disease. So what I did for today is I took this model and simulated 200 new data sets from that model, in all of my simulations, the null hypothesis was true. So I assumed for each individual person that they had a 4% chance of getting the diseases. So now we have 200 data sets, and in each of them we count how many persons have the disease. And this is what this looks like. So most of the time, as expected, you would get counts between 2 and 7. But sometimes you also see more extreme counts. For example, here you get some 0s and 9s and 10s. And these are light blue because these are values which will result in a significant test. But since I simulated in a way that the null hypothesis was true, these are clearly false positives. So that means sometimes tests become significant just by chance. These lead to false positives, which are a problem, and it has to do with the significance level. So when we're testing at a significance level of 5% and the null hypothesis is true, then we expect that 5 out of 100 tests would be false positive by definition of the significance level. The more tests we perform, the more false positives we expect. And if we perform 20,000 tests for 20,000 genes, we would expect a lot of false positives. So that means that we need some additional control to make sure that we found more hits than we would expect by chance. So I looked a bit into that data set, and if these authors had reported all the genes with a p-value below 5%, then this list of genes that they found would contain 1,500 genes, and only half of them would be expected to be true positives. And the reason to cope with, or the one way to cope with this problem, is just to use a stricter p-value cutoff and to lower the cutoff. And there are indeed methods that allow you to control the false discovery rate, which is the fraction of false positives that you would roughly want to have. And by this, they defines the p-value at which you can cut off. So by using this method, they reduce the list of genes to 80. And they know that they now can expect roughly 4 false positives within that list. And they say that they can live with that. So unfortunately, they don't know which ones these are, because otherwise you could kick them out and then you would have 0 false positives. So I hope that you learned something from that. So as a summary on the last slide, I thought I could give you some questions at hand that you can ask when you read a paper and you have some statistical results. What could you ask to understand better what you're dealing with? So the first question that you should have is, does the test answer the scientific question? And now this sounds super trivial, but indeed in my consulting sessions, I feel like I spend almost half of the time discussing this question, really. Sometimes it's not that obvious. Also, if a test was performed, you can look up the assumption that the test makes and ask yourself whether they are reasonable. So you learned today that the assumptions are always somehow wrong. So they don't have to be 100% perfect, but at least they should sound somewhat reasonable. Also try to get a look at the data points themselves, not only at the p-value. And if it's not the data points, at least an effect size should be reported, and you should ask yourself if this effect is big enough to have a meaning in the biological context. Also, if you are dealing with large data set, you should ask yourself whether there was a correction for multiple testing, and if there was no correction, then you should ask whether there was a rationale behind performing exactly those tests that are shown, or whether this was just a tricky way of avoiding the quality control steps. Okay, that was it from my side. So I think I would hand back to Anna.
Great, thank you, Sarah. So in just a moment, we will take a look at some of the questions. There have been quite a few. I think some of them have been answered already. Some are perhaps a bit too big for the scope of today, but we'll definitely look at some of those questions shortly. And if you have any further questions, do add them and we can take a look at those. Until then, I just wanted to share a couple of slides with you. So this was obviously particularly an introductory webinar, but Sarah has in fact put together an e-learning course which is available on the EBI training site. This is a much more practical introduction and goes into more detail on a lot of some of what she's talked about today, but a lot more beyond that. So if you're interested to learn more, I really do recommend you taking a look at this practical introduction. On to the next slide, please, Sarah. And I also wanted to mention that this is just one of our webinars, but we do run many. And in fact, this is the first of 2 this week. It may be interesting to some of you. We are having a webinar tomorrow around human missense variation with a new tool coming out of the EBI. So it's ProtVar. That's available on our website if you want to register for that. And then in a couple of weeks, the next webinar we have coming up is around cancer models. So again, maybe of interest to some of you. But as we mentioned throughout today, this recording and all our webinars are recorded and they are made available on the training site. So do go and have a look at what we do have in terms of recordings, because there is a lot there of all our webinars over the last couple of years. So I'm sure many of those will be of interest to you. And I think on to the next one, and that will just say, yeah, any questions? So we'll take a look at some of the questions now. So as I said, some of this may have been mentioned a little bit before, but one question we've had is around having lots of samples. So the p-value becomes artificially inflated when we have lots of samples. Why is that?
Okay.
Number of samples?
Yeah. No, I wouldn't say that it's artificially inflated. I would just say it becomes very low, because if you have lots and lots of samples, that means you're collecting more and more evidence against your null hypothesis. And I mean, artificially, I would say if you have samples that are not independent of each other, because this is also one very important assumption that we make in almost all of our models, that the samples that you're collecting are independent. So they are not, like if you collect data about students, if they are all from the same university, right, they will be somewhat similar to each other. So if you collect lots and lots of data points from similar areas or from similar conditions, they will not be representative for what you're looking at. But at the same time, by having so many samples, you're somewhat claiming that you have a lot of independent data points, which you don't have. So in this case, I would say it's artificial. In the other case, it's not artificial. It's just lots of evidence against the null. But at the same time, right, this should be another question then, how large is the effect? So if you get a low p-value, also please have a look at the effect.
Okay. Someone has just asked if you could clarify what the effect size is.
Yeah. Okay. So this is the rather general term. So in the example that we just saw, for gene expression, it would be the difference in average expression. But it also, in this example with the disease prevalence, it would be how much difference is the disease prevalence in the test group from what your assumed 4%. So the effect size is most of the time a difference between 2 conditions that you're trying to find out.
Okay, thank you. A few of the questions are asking a bit around which test to choose. Mm-hmm. So obviously it's difficult to go into depth on this right now, but do you have any guidance on trying to identify which tests are suitable?
Yeah, it's hard for me to make a general statement about this, really. So, I mean, if you're in biology, you were mostly dealing with what kind of properties your data have. So for example, you should ask yourself, is the data on a continuous scale? Then maybe it's a Gaussian or log normal distribution. Is it counts? Then we have a different scenario. We have distributions and tests describing count data, or sometimes we even have categorical data where the data fall into categories and not numbers and counts. So I think this is maybe the first step to make. Ask yourself what your data are showing, and then these are also terms that you can Google. It will maybe guide you to the correct tests. I hope that helps.
That's great. Yeah, I think that will certainly help. It's about understanding your data so that you can work out what to Google, taking that as a first step. Someone has mentioned about something they've seen recently regarding the use of p-values and journals not particularly appreciating, or some journals not saying that p-values are required. They ask if you have a view on whether p-values should be essential for papers.
Yeah. It's a difficult question and also a big question. So I can imagine that this depreciation of p-values comes somehow from them having been overused in the past. So maybe this is a counter movement to this mindset that some people have, that everything has to be proven with the p-value, and that the p-values are the ultimate truth, which I would agree with that they are not. Right? They are one piece of information that you have on the data, and I think in that sense they are useful. But I also don't see a point in putting p-values everywhere just for the sake of putting p-values. So sometimes a good visualization can be helpful. Sometimes you have p-values, and this I think I mentioned earlier on. They are there and they are comparing two groups, but comparing these groups doesn't help in any way in getting closer to the scientific question that has been asked. So I don't have a yes or no. I think you should apply some common sense, indeed.
Excellent, thank you. We have many more questions, so we're just going to answer a few of those. Because we don't have time to go into all of them in great detail, but just to cover a few more. So the next one is a simple question, but with a not particularly simple answer, I imagine. Is there a strict rule for the p-value cutoff?
No. So there are conventions. The most common one, I think, is to use this 5% p-value cutoff, and this will give 1 star, and everyone wants the stars. And then you have other lower p-value cutoffs that will give you more stars. But no, indeed, these are conventions, but they are arbitrary. Also, what I don't really understand is this need to get the p-value below 5%. So it's a cutoff at which you can, by some convention, put a star. But in the end, the p-values are not exact. I also tried to explain that, right? Because they involve assumptions that are for sure not 100% true. So I personally don't see a big difference between a p-value that is 0.049 and the p-value that is 0.051. I just don't find it reasonable.
Yeah, fair enough.
So both tell you that the data are rather unlikely under the null model.
So we've talked a lot about p-values today. Are there any other statistical values that you recommend to be thinking about or considering or adding into analyses?
I think that would depend a lot on the context. So I mentioned the effect size. I mentioned that in some cases you would have to adjust the p-values by multiple testing scenarios. I would always try to show the data themselves if possible. In huge data sets, of course, you have to bring it down somehow. But other than that, I would say it depends on the context and on the particular data that you're working with.
Okay, thank you. I'm just having a look at the questions. Sorry, trying to read at the same time. So somebody has asked, just trying to find it again. They're asking about means and standard deviations and medians, and they're asking, they say, when do we use mean and standard deviation, and when do we use a median or IQR? Is it based on the sample size?
No, not particularly. I would say it depends a little bit on the properties of the data. So the mean and the standard deviation can describe a normal distribution perfectly if your data are normally distributed. Then in this case, the mean and the standard deviation make absolute sense, but again, they are summaries of the data, and data are almost never perfectly normally distributed. So the median will give you the value at which half of the data points are smaller than that value, and in a lot of data sets, the median will be very similar to the mean. But especially if you're dealing with large outliers, they will greatly influence the mean and the standard deviation. So in this case, it can often be more useful to use the median, or there are even values that give you the peak of the distribution or something that are more descriptive for that. And in the end, it comes down, I think, if your data follow like an unusual distribution, I would say try to visualize that distribution somehow by a histogram or something. That will also give you further insight.
Okay, thank you. We have, I think, just time for a couple more questions, perhaps. So we have one here. So they're posing an imaginary scenario. So they have healthy individuals and diseased patients, and they want to develop a model comparing healthy and diseased patients. So they're asking how to define a sample size. So how many samples how would they work out the number of samples in which they need to
okay. Yeah, so this is a classical scenario where you are interested in power calculations or power simulations also. So you are probably interested in having a data set that is large enough so that if there is a difference between your two groups, then the test that you're applying would detect it, right? Again, we have conventions here for how large the power should be. So one very common convention is to say we want a power of 80% to get some clinical study design through. So that means if there would be a difference between the group, we want an 80% chance of finding it with the method that we have. So if you remember from the talk, I said that the p-value, and this depends on several things, and the power depends on the same thing. So we said it's easier to us to find a difference or detect a difference if we have a large sample size, if we have small variance within each group, and if we have a large difference in whatever we are comparing between the two groups. So if you wanted to calculate the number of sample sizes that you need, your calculation would depend on these values. It would depend on the difference that you're trying to detect. So if you have some kind of preliminary experiments or values from the literature, this would be great for estimating how many samples you need. Also, how much your values spread. The more the values spread, the more data points you will need to detect the difference, because it becomes a bit obscured if you have a high variance. And also the significance level that you're trying to apply. And the lower the significance level, the more samples you need to prove it. So these are the values that go into that power calculation.
Okay, thank you. I think that answered a few people's questions as well. So to finish a couple of broader questions, someone has asked if you have any tips beyond attending this webinar of how to get started with learning a bit more about statistics. Just to mention for everyone, I have added in to the chat the link to the practical introduction to biostatistics that I showed on the slide earlier.
Yeah, and unfortunately, I don't have so much to add to this. So I did my best in this biostatistics collection that we have at EBI to give some practical tips and also to get you started on programming in R. So I typically work with R. I think this is, if you want to work with biostatistics, is the easiest language to get started in. I also added a few more links on the slides further on. But beyond that, unfortunately, I don't know that many good books and good tutorials. There's also one thing that I maybe haven't added. There's also on the bioconductor page, I think they also have more tutorials. I learned about that recently, but I haven't really looked it up. So this is one further resource that you might want to look into. I think this is more on specific data types then.
Okay, yeah. So if you would like to learn more, I really do recommend you go and take a look at Sarah's online course around this. It is a very practical introduction, as it says, and we'll give you some ways of getting started on biostatistics in R. Okay, and just a quick question of, I'm pretty sure I know the answer to this one, but do you need to become a mathematician in depth to understand some statistics to work with?
I hope not. So I myself, I'm not a mathematician by training. I'm a biologist, and I think I will remain a biologist in my heart. And still, I think a lot of the questions that we're asking in statistics depend on common sense in the end. I think, well, it can be helpful if this is more your type of thinking, also to answer lots of these questions with programming and simulations. But I think some understanding of math is of course helpful, but I think you don't have to study math to apply reasonable methods to your data.
Good. I think that will come as a relief to many people, hopefully. But yes, to say, do take a look at that introduction as well, and the links in the chat. So I think maybe let's leave the questions there for now. As I mentioned throughout the session, both the slides and the recording will be made available. You should already have the link to the slide that's been added to the chat. But the recording will be made available by tomorrow, and I will send an email out to everyone who's registered. So don't worry, you won't miss that one. And it'll also be findable on the EMBL-EBI training site for anyone who would like to take a look at it. And then we'll also link to the other course from there as well. So you'll be able to find both of those together in one thing. So I think that will just leave me to say thank you very much to Sarah for joining us for today's webinar. I hope everyone learned a lot. I certainly learned some new things too. Do take that learning with you, but also come back to the recordings as you see fit. And thank you very much to all our attendees and everyone who's asked your questions and joined in with the chat. I'm sure it makes it nice for everyone to be able to see that we're not learning about this alone. We're all learning new things together. Just lastly, want to say, do check out the other e-learning that's available on the EMBL-EBI training site, as well as the upcoming webinars and the recordings. So I'm sure there's lots more for you to learn available there. Great, well, I hope to see you all at a webinar in the future, and thank you.